Begin to look at the muscle physiology. Start off today by taking a look at the structure of the muscle cells themselves, so a little bit of a review of the anatomical structures that we have involved in skeletal muscle. As skeletal muscle is the basis for what we'll talk about as far as the muscle physiology goes. This is where we'll begin our journey in a sense. So essentially kind of like muscle, similar to that of bone, it's kind of packaged into smaller and smaller units. And so things are getting smaller and smaller as we go from the entire muscle. So we have our muscular organ itself, whatever that muscle happens to be. It's then bundled up into smaller packages called fascicles. And so those fascicles contain many muscle fibers. Those muscle fibers are the individual cells themselves. Inside a cell, those cells are then made up of myofibrils. Those myofibrils contain kind of bundles of proteins that are our actual contractile proteins. So we have things like our myosin and actin. Myosin is referred to as our thick filament. Actin is our thin filament. The actin filament, the thin filament, also has some regulatory proteins that are associated with it as well. And so we package this up so that we're moving from our whole muscle that we have. And when we look at the structures within that given muscle that is there, we can see the individual fascicles that are within the muscle itself. And so we have these whole bundles that are there. And inside those whole bundles, we have individual muscle fibers. And so we have individual cells that are inside. And within those individual cells, we then have our myofibrils. And inside the myofibrils, we then have our myofilaments. And so we have the actual muscle proteins themselves that are going to do the actual work of the muscle at hand. So when we look at that single cell, we have the general structures that we can see on the outside of the cells. So we have a plasma membrane, or the sarcolemma. So that's the outer covering of the cell. And then inside there we have our bundles of our myofibrils. So this one's been cut in half here. And that myofibril has the individual proteins. And so we can see our thick filaments, the darker bands, and then the thin filaments, the lighter bands inside there as well. Other things that we have inside here, we have our SR, or our sarcoplasmic reticulum. That sarcoplasmic reticulum kind of surrounds the myofibrils. This is our endoplasmic reticulum for muscle. We'll find all of our calcium stored within inside there. That sarcolemma itself continues on deep into the inside of the cell. It kind of invaginates and creates little tubules. So we have what we call our transverse tubules, or our T tubules, that run along right next to the sarcoplasmic reticulum. And as that sarcoplasmic reticulum then is stimulated by an electrical impulse coming down from the sarcolemma through the T tubules, then that sarcoplasmic reticulum can release all of the calcium that is stored within. And so we have the ability to very quickly flood the inside of the cell with the things that it needs. Skeletal muscle is also multinucleated. So we have a nucleus over here, we have a nucleus here, we have a nucleus here, providing for all of the information needed to make more and more of these proteins, make more myofibrils inside there, so that we have a larger and larger muscle when we need to. Supplying that muscle, we have mitochondria. So many mitochondria inside the cell. Depending upon the type of muscle cell is going to dictate how many of those mitochondria we have, going to dictate how extensively filled it is, all based upon the needs of that muscle, what it's supposed to be able to do. So looking closer at our filaments, we have our thick filament here. The thick filament itself, remember, is made up of a tail and a head. The tails themselves, as they wind into the thick filament, we take two myosins, and we're going to actually twist two of these myosin tails together, and that's going to leave two heads extending out from it. And so there would be another head over here as well there. And as those come together, they have a little bit of a hinge point in them. This is going to allow the myosin to essentially reach up, grab onto our thin filament, and then pull it over to the other side. And so it's going to allow it to move and pivot and allow for us to generate some force within the cell itself. And so that's going to help to generate the force needed for muscle contraction inside the cells. The couple other things we can see on this particular slide, we have some regulatory proteins as well, or some associated proteins within the cell. We have titin filaments, and then we have our M line, which is created by our myomesin. Myomesin is going to help to hold that cell, remember, and hold all of those thick filaments in place. So we'll go up and connect to another one, go down, connect to another one, all within that myofibril. Titin filaments are going to, as it appears to look there, it's kind of like a spring. This is going to help with the stretch of a muscle. As we stretch that muscle out and lengthen that muscle, it gives it its rebound to the muscle. Our thin filament has some proteins in addition to the actin protein as well. And so all the little red balls on here are our actin proteins. Essentially two different ways to look at it. One is the individual balls themselves, those are the G-actin. The entire bundle as they're strung together, kind of like a string of pearls, something like that, we have the F-actin that is there. The regulatory proteins are our troponin and tropomyosin. These two are going to act kind of like a lock and a door. And so we have a door here, and we've got a little lock on the door. That little lock are troponin, so the lock is the troponin, the door itself is the tropomyosin, and on the other side is our actin with its little binding site that it has on it. And that little binding site is looking for myosin to be able to grab onto it. In order to get to the actin, we have to open up the door, which means that we need a lock for our key. If we don't have a lock for the key, then we're never going to get through the door and into the other side where we have our actin. So the proteins that are there, oops, I undid my door there. Our actin and our myosin and our troponin and our tropomyosin are all integral to the process of allowing us to have muscle contraction. And so on here, we can see the little yellow portions on the red balls. Those are the binding sites. And normally when a muscle is relaxed, all of this green tropomyosin covers over those binding sites. So that's our door. It's blocking our way to the actin behind the door itself. But if a little calcium comes along and binds to our troponin, that's going to change the shape of that troponin. It's going to move the troponin, which then moves the tropomyosin, and our door then opens up. And we can see our actin binding site and the actin behind it. And now it's ready for a myosin head to come in and bind to it. Now it can get to it. It has the ability to bind onto that binding site, and we can now have muscle contraction. So the proteins themselves that are there allow for that kind of process to occur and are going to be integral to getting our normal muscle contraction that we want. All of those thin and thick filaments and all of the regulatory proteins that are inside there are all within what we call a sarcomere. A sarcomere is divided from one Z disc to another Z disc. And so a single unit of overlapping thin and thick filaments. And so the borders of it are the Z discs and the Z discs. And so we have these proteins that are on the end there. The myosin are attached to those Z discs, and one set of myosin, or I'm sorry, the thin filaments, the actin makes its way towards that sarcomere, and this makes its way towards that sarcomere. And so one side has sarcomere of the sarcomere has actin pointing in from either end. That's going to be our actin here waiting to be able to bind to our myosin head. And so the myosin head, now we can see on those tails how they are twisted together there. And so two of them are twisted together and put together, and we have the two heads sticking up from there on our thick filament. When muscle contracts, then those Z discs are going to move towards the M line. They're going to end up moving towards the center of the cell, that myomesin there, and those filaments are going to start to overlap one another. And so the heads are going to bind to all of these little binding sites on here and start to pull the thin filaments in towards the center of that sarcomere. And then this one's going to be doing the same thing. And so it's pulling its thin filaments in towards the center as well. And we get that contraction that is happening inside the cell. The sarcomere here, we can also see how the thin and thick filaments are aligned. And if you remember back to microscopically, we saw areas of dark and areas of light. And so dark over here, light over here, and those allowed for those striations that we see on the muscles themselves. We have the dark areas and we have the light areas giving that striated appearance to the muscle cells themselves. Some other things in terms of regions that we can see in our sarcomere itself. So we said that the sarcomere goes from Z disc to Z disc, and so that's one entire sarcomere that is there. Within that, we have some areas that are going to change in terms of the regions in a sense. And so bounded by the very end of the thick filament and the very end of the thick filament, we have the A band. And so it goes from the very edge of the thick to the very edge of the thick. In the middle of that A band, we have this area right here, which is our H zone. And so the H zone is the area where essentially we have only thick filaments. And so there are only thick filaments in that area. And depending upon whether the muscle is contracted or relaxed, the amount and space of that H zone is going to change. It's also going to change for the I band. And so the I band is kind of the counterpart to the H zone. The I band is the area that goes from where we have only thin filaments. And so it's going to go from this particular end of the thick filament all the way over into this other sarcomere, into that other sarcomere, and up to the edge of the thick filament as well there. And this whole area here, where there's only thick filaments, this is our I band. We again can see the tightened filaments, how they're attached to the Z discs. And so they align themselves up. Their curly little springs are on there. And so as we stretch that muscle out and pull that sarcomere into a lengthened position, those are going to stretch out and help with rebound of that muscle, get it back into that area where it is no longer stretched. Again, we can see the M line lining up all of our thick filaments inside the cell. And then we have the last area is the zone of overlap here. And so this area where we have both thin and thick filaments. And so the I band and the H zone are going to essentially disappear as a muscle contracts, and the zone of overlap is going to increase. So it's going to increase in its size until the thin filament is essentially all the way there. And so then our zone of overlap would essentially stretch the same as our A band as we contract that muscle. And so that's going to essentially elongate. The H zone and the I band are going to decrease in their size. And we'll talk about the actual muscle contraction the next time around.